So in our study, we looked to sort of trace the evolution of CLL towards Richter's, and we definitely found some key features of Richter's transformation. After we did our clonal deconvolution, which allowed us to determine the genetic alterations that were actually happening at the level of the Richter cells themselves, we found several novel drivers that were associated with Richter's transformation. We also found that there was numerous copy number alterations that were also happening, the majority of which were happening at the time of Richter's, also in the Richter's clones. Other features of transformation that we found that were quite prominent is we found whole genome doubling in about 15% of our cohort, and also frequent altered genomic events in terms of chromotripsis and cateches. One of the other things that we were able to do in our study is to look at serial samples for characterization and follow clones over time as the CLL evolved towards Richter's. And so this confirmed prior findings that had been previously based on B-cell receptor sequence or IGHV sequencing, that Richter's can exist both as clonally related to the underlying CLL. That means it evolves out of a pre-existing CLL subclone, and the majority of our discovery cohort, 86%, fell into that category. Richter's can also exist as clonally unrelated, meaning that it doesn't come from a pre-existing CLL clone and is actually a distinct malignancy. And we found that to be the case in 14% of our cohort, where the CLL and the Richter's didn't share any somatic genetic evolutionary history.